Guys, so this is the video and structure of DNA part. And if you look right here, you can see that we have our learning target right here. Describe the structure and function of DNA, which is what we're doing today. And then later we'll do RNA. So that is the learning target for today. By being able to describe the structure of DNA and knowing about the structure of DNA allows us to know how DNA works. How does it transfer our genetic information? How does information get copied? How does our cells know which proteins to make? So to do all of that, you have to know the structure of DNA. And so that's why we're studying it. So if you look, we're going to kind of work backwards right here, but here is our twisted ladder. That's what we know DNA looks like so far. And so to study it, we're going to untwist it. That's called a double helix. That's a fancy name for twisted ladder. And so if we untwist it, we're going to see that it looks like a regular ladder, and you have sides of the ladders, and those are made by a deoxyribose sugar. And then they're connected by phosphates. So those are the sides. And then the steps of the ladder are made by nitrogenous bases. And you can see two different nitrogenous bases will actually bond or connect together to make the steps. And so if we zoom in even more, the deoxyribose shape, that sugar, that molecule is shaped like a pentagon. So we can see the blue sides here. Well, that deoxyribose sugar is shaped like a pentagon, and then it connects with a phosphate to another deoxyribose sugar molecule, and then that connects to a phosphate, and that connects to a sugar, and then it's so forth. So that is the sides of the DNA. And what's weird about one side of the DNA will connect in one direction, like you see your pentagons pointing up. And the other side of the ladder of the DNA is bonding in the opposite direction. It goes down. So they're like cattywampus. One bond's going up, one bond's going down, and that is a little strange and can make for copying issues and things like that for DNA. So it's a little bit wonky. If you look in the middle, there we go with our nitrogenous bases like adenine and thymine, guanine, cytosine. Oh, here's another adenine and thymine. So we're noticing that only certain nitrogenous pairs will hook up. And that's how it is. Adenine only wants to hook up with thymine. Cytosine only wants to hook up with guanine. They're not going to mix. And if they do, you got a problem. So this is kind of unique to DNA as well. So you have the steps of your ladder with your nitrogenous bases. So this is kind of a general picture, zoomed in a little bit, zoomed in a lot of it. And so that's what that looks like. So now if we look, your notes are going to be filling in the blank. And you have done this before in previous. Same kind of thing. So you have a word bank at the bottom. And I know what I want the first one to be. And so I am going to just demo this briefly. And so I know that I want the first word to be the sides. And so if we look at that first word, and so this is what you guys will do. You'll go in and fill in the blanks. You can either write it or copy and paste it. But in this case, I'm going to put in the word sides. So the sides of DNA, the DNA ladder, are made by, and then you guys will fill in the blank here, deoxyribose sugar. So the sides of the DNA ladder are made by deoxyribose sugar, and they are connected together by phosphates. So I'm going to pause right here, and then I will finish going